So we're Dr. Arevelo's lab, and we did this study on dog evolution.
So just to kind of give an introduction, so our lab uses dog DNA samples to better understand evolution and genetics. So dogs serve as a good model organism because their genetic basis of disease, morphology, and behavior is similar to that of humans. Dogs are also a convenient model organism because it's easy to get DNA samples. So our DNA samples are actually mailed to us, and they come as either cheek swabs or blood samples. And additionally, dogs, because of the way that they're bred, it's easy to get samples from entire families and scale across different generations, which is really helpful for genetic studies. So our lab focuses mainly on the hoverwort. So in figure 1, you can see a picture of the hoverwort puppies. They're a breed that originated in Germany. When they grow up, they look like either German Shepherds or Golden Retrievers, kind of. And so what we do is we use molecular and statistical tools to analyze our data, and we want to learn what makes dogs dogs. So we're looking at different phenotypic traits like height or ear shape, as well as disease. So our objective, what we do is we use single nucleotide polymorphisms, or SNPs, which are basically just variable genetic markers. And so we want to use these to look at the relatedness of different dogs, as well as link different loci and phenotypic traits. So one trait that we look at in detail is a mutation in SOD1. So this gene has been associated with a disease called degenerative myelopathy, which is basically ALS in dogs. And so we also have determined a unique insert in this gene in the hoverwort breed that we look at that we're examining as well. So, and our materials and methods.
So as you heard Megan say, we get sent a lot of samples of various dogs. What we do with these samples is we extract the DNA from them using different enzymes. And then we take these DNA samples and run them through a machine that's able to genotype hundreds of these different SNPs. Then we use our statistical software to analyze all of the information that we gather, and it compares dogs, their different genetical information. So we looked at over 300 hoverwort samples, and as you can see, we have a chart here with all these bars and colors. So each vertical bar represents a different dog, and you can see that all the red ones are all related genetically, and all the blue ones are related genetically, and some of them have a mix of different colors. So what this shows us is which dogs are from which place and how closely related they are. We can see in the first 2 rows of this chart that there's mostly red and blue bars. What that's telling us is that these are mostly North American dogs, and they're more closely related to each other than they are to the European dogs, which compose the rest of the chart. As you can see, there's more colors in the rest of the chart. So that suggests that there has been a genetic shift when these dogs came to North America, which makes sense because only a small sample of the hoverworts from Europe came over to America. And you can see in the tree next to it that, for example, each color corresponds with a different color in the bar chart. So the 1 is the furthest away from everything else. It corresponds with the red ones, which makes sense because these are mostly North American dogs, which seem to be separated from most of the European dogs. What we have below that is a map of North America and Europe. So each color dot represents a different group, and these groups seem to be very geographically centered. So you can see in France, there's a green dot, and we have in America 2 pink dots on either side of the country. In Texas, there's a green dot, which shows that each location has different genetic variability. And now we can move on to our conclusions.
So when we were continuing to look at genetic variability, additional tool that we used was gel electrophoresis. So if you look in the upper right-hand corner at figure 5, for our digest, when you look at the PCR, the uncut top bands are what we would expect to see, whereas the middle bands are obtained when you do the digest, and just kind of shows the original band begins to break down, And this process allows us to know if an individual is homozygous or heterozygous. And so the alleles that we would consider here are G and A, with an A allele showing a predisposition for the disease. So just some things to consider. So our wild type is approximately 270 base pairs, which would be the upper bands. And we know that the A allele will not cut. So overall, this just helps us show genetic variability. So before COVID-19, we were actually having some issues with our amplification process because our bands were showing up a little bit higher than expected, but we started to get that under control. So when we return to school, we plan on cutting out these bands and sending them out to be sequenced. And I'm going to pass it off to Katie to talk about our insert region that we found as well.
So as Jordan just mentioned, and also Megan mentioned before, we've been studying like an insertion region. So this region, it corresponds to the Italian paper that they have done. They have also done this kind of study on, and this in specific insertion, it's specifically related to the Hobelwart dogs that we've been studying. And it shows that this insertion alters the screening for the PCR, which overestimates the disease alleles. So it's giving a lot of people issues with actually diagnosis. So that's why we've been studying this insertion region to see like whether if it's really genetic or if it's just from the exon 2 being duplicated and reinserted.
And just to kind of go over our conclusions, we found that like the clusters mostly reflected geographical origins, and to improve genetic variability, it's important to breed dogs that are from different countries. Our multi-generational samples also showed the passing of traits down familial lines, which includes genetic insertion observed in SOD1 gene that Katie mentioned, associated with DM. And by assessing local genetic variability among Hobelwarts from different localities, we'll be able to advise breeders on the best mating practices and avoiding intergression, etc.
Thank you both so much. Just following up on that, can you talk a little bit more about why genetic variability within these dog breeds is so important?
Megan, you're muted.
Sorry, was muted. With dogs, just the way that they're bred, they're basically an artificially selected breed, and the breeding patterns with humans can cause like a lot of problems. So people who know stuff about dogs often know that with purebred dogs, there tends to be a lot more disease because of all the inbreeding and stuff that occurs. So certain dogs are known, like German Shepherds are known to have hip dysplasia. So it's important to evaluate these different breeding patterns to understand how this can lead to disease and how we can kind of prevent disease from occurring.
Wonderful, thank you. It's really important and interesting work you guys are doing. Thank you for sharing it.
Thank you.
Thank you.